Knowing the structure of RNA is also very important. The main differences between RNA and DNA is that it has ribose as the sugar instead of deoxyribose. It has uracil, which we'll call just the U oftentimes, is going to replace thymine. It is primarily going to be single-stranded. There are some exceptions to this. Some RNAs do form double-stranded regions as they fold into different secondary structures. Also, some viruses have a double-stranded RNA genome. We'll talk more about some of the exceptions to this rule, particularly when we talk about things like gene expression, because they can really help to regulate gene expression. So if we were to look at the structure of this, again, it's primarily going to be single-stranded. You're still going to have a phosphate sugar backbone. And you can see here the nitrogenous bases, the difference is you're going to have a uracil instead of the thymine. There are three main kinds of RNA that we will talk about, and these are all important during expression of genetic information. They're all really, really important for transcription and translation. Messenger RNA, or mRNA, is a copy of the DNA. It originates in the nucleus, it leaves the nucleus, and can go out to the cytoplasm. Ribosomal RNA, or rRNA, will make up the ribosomes. These are very important for protein synthesis. They're going to act as basically little machines, and the messenger RNA is fed through them, and the ribosomal RNA will basically read and help to interpret this into a protein structure. The tRNAs, or transfer RNAs, are important for bringing amino acids to the ribosomes. Remember, amino acids are going to be the subunits of proteins, and so we need to bring in many, many different amino acids so you can have your growing protein chain. We'll talk more about these when we talk about transcription and translation in a little bit later. All three of these, though, originate as complementary copies of one of the two strands of DNA during transcription. There are several other types of RNA, and again, we'll talk more about those later. Telomerase RNA is in Chapter 11. It's involved in DNA replication at the ends of the chromosomes. Small nuclear RNAs, or snRNAs, are important for processing mRNAs, and that's in Chapter 13. Before an mRNA can leave the nucleus, it needs to be processed in several different ways. And then lastly, antisense RNA, microRNA, short interfering RNAs, they're all involved in gene regulation, and we'll talk more about those when we cover Chapter 17.